Let me start with something that happens very often in generic drug development. Everything looks perfect, assay is fine, impurities are controlled, and stability data is acceptable. But during submission, one simple question appears. What about residual solvents? In this short case study, I'll show you how one generic drug project faced a serious ICH Q3C compliance risk and how the issue was solved using the right analytical strategy. Hi, I'm Raghuvar Singh Parmar, Director, Scientific Affairs at Resolve Mass Laboratories, Canada. Resolve Mass Laboratories stands at the intersection of analytical excellence and complex drug development, helping pharmaceutical teams overcome critical challenges in peptide, impurities, and regulatory pathways. Why Q3C compliance matters? Residual solvents are not just processing chemicals, they directly affect patient safety, especially when medicines are taken for long periods. This is why ICH Q3C provides globally harmonized limits. For generic drug companies, this is not optional. If residual solvents are not controlled properly, the entire submission can be delayed or even rejected. ICH Q3C solvent classification. So let's look into the classifications of the solvents. So ICH Q3C divides solvents into three main categories: Class 1 solvents, Class 2 solvents, and Class 3 solvents. Class 1 solvents should be avoided completely because they are carcinogenic. Class 2 solvents like methanol and acetonitrile are commonly used but must be strictly controlled. And Class 3 solvents are considered low risk, but they still require proper testing and documentation. Understanding these classes early helps avoid major regulatory problems later. Now, let's talk about difference between ICH Q3C and USP Chapter 467. Many teams get confused between ICH Q3C and USP 467. The simple way to understand this is ICH Q3C defines the limits, while USP 467 general chapter defines how the testing should be done, especially for United States submissions. So in real projects, both must be followed together. Ignoring this difference can create serious compliance issues. Now, this is one of the case study that I want to discuss here. In this project, the synthesis process used acetonitrile and methanol, both class 2 solvents. The internal testing was completed and everything looked acceptable, but there was one major problem. The method was not sensitive enough to detect low levels of the solvents. This created a big regulatory risk because the product could still fail even if internal testing looked fine. Another case study is about resolution and results. So to solve this problem, a high sensitivity headspace GC method was developed and validated. After improving the method, the detection level went below 10 ppm. The final results showed acetonitrile at 215 ppm and methanol at 1100 ppm, both within the acceptable limits. These helped the project move forward successfully without regulatory issues. Typical ICH Q3C solvent limits. Some solvent limits appear again and again in generic drug projects. For example, benzene has an extremely low limit. which means even small concentration can create problems. Acetonitrile and toluene also have strict limits, while ethanol is considered safer but still needs proper control. Knowing these common limits early helps in better method planning. PD calculations and updates. ICH Q3C limits are based on something called PDE, permitted daily exposure. These limits are calculated using toxicological data and safety factors. This means the limits are not random, they are scientifically designed to protect patients. Understanding PDE also helps team make better decisions during process development. Expert testing services. This case study also shows how important method sensitivity and proper validation really are. High sensitivity testing, fast turnaround time, and audit-ready reports can make a big difference during regulatory submission. When the testing strategy is correct, the entire project becomes much more smoother and faster. So the key takeaway from this case study is simple. Residual solvent testing should not be treated as the final step. It should be planned early in development. If the right analytical method and regulatory strategy are used from the beginning, companies can avoid delays, reduce risk, and achieve faster approvals. If your program demands precision, expertise, and regulatory ready data, contact Resolve Mass Laboratories to support your complex drug development journey. Thank you so much for watching this session, and see you in the next one. Goodbye for now.